Hello, I'm Martin Blaser. I'm the director of the Center for Advanced Biotechnology in Medicine, or CABM. Today, I have the pleasure of speaking with CABM resident faculty member, Dr. Ian Oldenburg. He is an assistant professor in the Department of Neuroscience and Cell Biology at Robert Wood Johnson Medical School. Hi, Ian. How are you doing?
I'm good. How are you today?
Great. So Ian, I just want to ask you a couple of questions so we can start a dialogue.
Sure.
Can you tell me about your education and your early career before you came to CABM?
Sure. So I did my undergraduate work in Carnegie Mellon, which is in Pittsburgh, Pennsylvania. I majored in biology there, and I worked in a couple different labs over my undergraduate time, but the longest stint was my, basically an entire senior year working with Justin Crowley studying the visual system. So he really wanted to learn a lot about how the visual system develops and the changes that happen over development of the visual system. From there, I went to graduate school at Harvard Medical School, and did my PhD work with Bernardo Sabatini. I studied the striatum and the basal ganglia in general, and what I was most interested in the basal ganglia was the control of movement. So I wanted to know how the different brain areas talked to each other, how the motor cortex talked to the striatum and vice versa, and how this sort of central brain region was able to talk to so many different things and really have strong control over these reentrant loops. So activity from the striatum talks to thalamus, talks back to cortex, which then talks to striatum. And so you get this really complicated reverberating activity that I thought was quite interesting. Then from there, after finishing in my work in Bernardo's lab, I did a postdoc at UC Berkeley with Hillel Adesnik, where, having looked at large populations of cells and, many cells firing simultaneously, I really wanted to get into the weeds of how individual cells might affect their neighbors. And so I worked with Hillel and several collaborators in Berkeley to develop techniques that allowed us to stimulate individual cells without stimulating their neighbors, to actually answer questions about the visual cortex. So it tied back into some of the work that I had been doing in my undergrad time, looking at the visual cortex, but using much more advanced microscopy techniques. So.
Right. That's terrific. So I know that you have recently come to CABM, and the empty bookshelves behind you are kind of evidence of that. But
Yep.
Tell us, what brought you to CABM? Why did you come here?
Oh, well, there are a lot of reasons. I mean, number one thing is, when talking to everybody at CABM, I saw, first of all, a huge breadth of skills. There were people who were doing a lot of very different things in very different labs, and everybody was talking to each other and learning from each other and, expanding their horizons a little bit. And yet, since at least my PhD, I've really enjoyed these cross-disciplinary collaborations. So, in my PhD, I worked with various material scientists to develop new tools. In my postdoc, I worked with optical physicists and molecular biologists and several other different scientists. And I really love when you get people with different skill sets coming together, tackling the same problem, moving things forward, because you come up with solutions and even questions that you never would have come up with by anyone yourself. And it seemed like CABM was really the only place that I interviewed that sort of represented this as a physical body, right? Where we have people doing very different things sitting next to each other. And so I can't tell you right now what collaborations I want to do, but I just love the idea that everybody is here, and new ideas are going to pop up, and we're going to travel down ideas that we wouldn't have otherwise even thought of. So
Great, fair enough. So in that sense, then maybe you can tell us, what's the main focus of your lab as you extend it? What are the key questions you want to address, at least initially?
Yeah, so that's a good question. So the idea for my lab is to sort of take take aspects of different parts of my career to date. So I'm centering the questions around understanding how the motor cortex works, which is something that I was sort of peripherally interested in grad school. I was interested in the basal ganglia and how the basal ganglia talked to the motor cortex. Well, now I want to get into the details of the motor cortex itself. And I want to use techniques that I was developing in my postdoctoral work to be able to stimulate individual cells to be able to see their effects on networks and on behaviors. And so this all comes together addressing the question of like how neural activity actually drives behaviors and actually translates into behaviors. And so the way I usually go about describing this is that we know from decades of work that these rich patterns of activity in the motor cortex are associated with different actions. And you can see certain cells like certain actions, certain firing patterns are correlating to different actions and different movements. But taking the next leap from saying this activity is associated with that versus this activity drives that or shapes that activity in a different way, it's actually a really hard question to answer, but sort of necessary if you want to fully understand your system or build a really good neural prosthetic that knows who to talk to and who to listen to. And the only way you can get at these causal questions is if you actually have a very precise manipulation. So these tools that I was developing in my postdoctoral work allows us to go in and rewrite, activate individual cells or rewrite patterns of activity that we saw and say, okay, are these patterns the ones that are driving those actions, or is it different, or is it part of this pattern matters and part doesn't? And so that domain of understanding how neural activity gives rise to action is the central question of the lab.
Great. So within that, can you tell us about one project that's particularly exciting to you that you're raring to go?
Yeah, so that's a good question. So I think the project that I'm most excited about right now, and I might change my mind in a week or two, but what I'm most excited about right now is actually looking at how different brain areas talk to each other in a little bit more detail than you would want, than you would otherwise normally have access to. So we have our microscope that allows us to stimulate many cells. You can stimulate pretty much arbitrarily in a very large field of view. And then we can put an electrode in a brain area that we know your primary, your motor cortex talks to. And so, for example, and the first thing I would try out would be connecting the motor cortex to the striatum, because as I said, I know a lot about that connection. And then we can stimulate individual cells and in different patterns in motor cortex, and we can see what patterns of activity best drive the striatum. Now, normally, because we have cellular resolution and activating individual cells, you can imagine this is an incredibly complex parameter space. Think it's 2 to the power of 200, maybe, different combinations of cells that might be the combination that best activates your cell. And this is a totally intractable problem at first glance. But in reality, we know that this is a very common machine learning scale of problem where you might have this degree of variability. So we can use lessons that we've learned from the machine learning world to follow gradient descent down to find what patterns of activity in the motor cortex are the ones that are best driving striatum. And we can, borrow ideas and techniques from different fields and actually come to a pretty straightforward answer. Now that will only get you the answer of which cells talk to, which cells are best driving striatum, which is sort of a what answer. And then the sort of fun part becomes translating that into a why answer. Why is this true? And that's where all of the fun biology of tracking down who are these original cells, when do they normally fire and how does that relate? But this is, the first you solve the what, and then you come up with the why, and then that's science at work, right?
No, that sounds very exciting to me. I'm ready to sign up to become your first postdoc.
Well, I'm hiring, so please come on over.
Can you tell us how you interact with postdocs and students?
Yeah, so my my philosophy is that, the people in the lab are the most important thing. And so my job as the lab head, as the PI, is to clear barriers for the people that work in the lab. And, sometimes that's, a scientific problem. We need to talk through an idea or, flesh out an idea to make it great. And sometimes it's an administrative problem that having a PI email someone saying this just needs to work is what it takes to get a project done. And whatever it is, that's my goal, is to clear objects, help people find what they're looking for. It's not for me to decide what your goals are. That's, but it is for me to help you achieve whatever that goal is. So I really, I think the lab isn't a space, it's a group of people. So it's important to support those in whatever means I can.
Great, great. So now that you've come to Rutgers, what school are you in and what departments are you in?
Yeah, that's a good question. So I'm in the Department of Neuroscience and Cell Biology at the Robert Wood Johnson Medical School, and also a secondary appointment in the Department of Neurosurgery.
So you're planning to do brain surgery on people?
Not on people, no, but we do work on mice and do manipulations there. So
what do you hope to benefit by being in the Department of Neurosurgery?
Well, I think in the farther term plan, there are actually a lot of ideas that could flesh out. As we develop new ideas on how to use this bidirectional communication, where we're writing information into the brain, there's a lot of hope that this will create a next generation of neural prosthetic. And so there's a lot of technology development that hopefully I contribute some to, and other labs will contribute to, but all of the theory and the ideas that I'm working on are directly applicable to creating better neuroprosthetics, specifically neuroprosthetics that don't just read out information and turn it into movement, but send information directly back to the brain. And so this is, what neurosurgery departments are many are all about, and we have a lot of opportunity to collaborate in the
Right.
future. So.
Right. So now I'm going to ask you my last question, which is a little odd question. So because you're just starting your career as a faculty member. So imagine it's the end of your career, and I'm writing your scientific obituary. What would you like to be most remembered for in your career? Where do you want your career to go, so that they'll say, oh, well, Ian did the following?
Well, first of all, I definitely want to be remembered for the people who passed through my lab. I think I take mentorship really, really seriously. And, I absolutely hope that anyone who's writing my scientific obituary, the first thing that they think about is, the people who came through and their experiences and their success in their own careers. That would make me happier than any specific scientific finding. Now, I'm very partial to this world of multiphoton optogenetics, the sort of broad class of techniques that I've been working on for a while. I have set up a conference around it, and I've been championing and trying to bring it to be more and more mainstream. So in terms of a large field and trajectory that I hope stays part of my career and my life for a long time, I want to be known for being one of the major voices in the multiphoton optogenetics world.
Great. Terrific. Well, Ian, it's great talking to you, and good luck as your career unfolds.
Thanks. Thank you. My pleasure.